Today we're going to start our unit on DNA. Hopefully you guys already know a little bit about DNA from your middle school science class, because DNA is really cool. Your DNA codes for who you are and how you're made, what color hair you have, whether your hair is curly or straight, what color eyes you have, the exact shape of your liver and how well your liver functions. It's everything that is you. Today we're going to start with a little bit of background information. How we discovered the shape of DNA, how we discovered that DNA is in fact your genetic material, and then some vocabulary that we're going to use to describe it. So let's start with how we discovered that DNA was actually the genetic material. Because way back when, before we even knew about DNA, remember, we were at the stage where we could see inside of cells, and we were understanding that there were these different things inside of them. And we knew that there was something inside the nucleus. There was this squiggly stuff in there, but we didn't know exactly what it was or what it was for. We weren't at that point yet. But we did know about proteins. So there was a huge argument between the scientists of the day of what our genetic material was. Was it proteins? Or was it that squiggly stuff inside the nucleus? Which one was the code for making you, you? And so we had a couple of scientists, Hershey and Chase, who finally developed an experiment that proved once and for all which one of those two things was the genetic material. They did this experiment in 1952. And they used two important things to help them out. One, they used a bacteriophage. This is one of your word wall words. A bacteriophage is a virus that will only infect bacteria. So it's safe to work with in the lab because it doesn't infect humans. It just infects bacteria. And remember, viruses are very simple because they only contain two things. They contain proteins and they contain DNA, the end. So it's perfect. They contain the two parts that scientists were arguing over what's our genetic material. And that's all they contained and nothing else. So this would answer that question. The other thing they needed to use was a radioactive marker. This is an element that you can attach to something that is normally not attached to. And remember, things that are radioactive, these elements decay and they release huge amounts of energy known as radiation. And we can detect that radiation on X-rays and using different radiation monitors. So here is an example of a bacteriophage. This is called a T4 bacteriophage. So Hershey and Chase did two experiments. One to test whether protein was the correct answer, and one to test whether DNA was the correct answer. They did protein first because they thought that protein was the right answer. So they used a radioactive sulfur because sulfur will attach to proteins chemically, but it will not attach to DNA because it doesn't match up chemically. So they used a radioactive sulfur, and they soaked the bacteriophage in the radioactive sulfur so that it would stick to all the bacteriophage's proteins. And then they allowed the bacteriophage to infect bacteria for a while. And then they tested the bacteria to see if it would glow with radiation under microscope. And when they did that, they found that the bacteria weren't glowing, that the protein did not get injected into the bacteria. So since there was no protein injected into the bacteria, protein can't be the genetic material. So they ran trial number 2, where they were testing to see if DNA was the genetic material. They used a radioactive phosphorus because phosphorus will chemically attach to DNA, but it will not chemically attach to protein. So they did the same thing. They soaked the bacteriophage in the radioactive phosphorus for a while, and then allowed the tagged bacteriophages to infect the bacteria, and then they tested the bacteria, and they found out that the bacteria were glowing with the radioactive phosphorus because the virus had injected its DNA into the cell. So because of this, they were able to say, well, what the virus is injecting is its genetic material because it's teaching the cell how to make more viruses, and what was injected was definitely the DNA because that's what we detected. So genetic material is your DNA and not proteins. Okay, here's a picture representation of each trial. Okay, you'll see in this first trial, this is where we used sulfur. That's why the proteins are red. And remember, we've already learned about how viruses infect. They attach to the outside, inject their genetic material into the cell, and the rest falls off. Well, the rest that was falling off was their protein. That's not what was being injected. Okay, trial 2, the phosphorus labeled the DNA, so the DNA is red. Okay, and that is what they injected into the cell to take over. Okay, if you still don't understand this experiment, pause the video and call me over, and we'll talk about it one-on-one. So now that the scientific world knows that DNA isn't that the genetic material that codes for how to make you, they turn to discovering the structure of it. So they were able to break down the chemistry, and they knew the chemicals that it was made out of, the carbons, the hydrogens, the oxygens, the nitrogens, but they just didn't understand how they came together into their shape. They didn't understand that yet because microscopes weren't good enough to look that close to be able to see that structure. But then there was a scientist named Rosalind Franklin, our first female that we get to learn about. And this was in 1952 as well. A lot of DNA work going on during that time period. This is a picture that she took of DNA. And the moment she took that picture, she sent it out to the scientific world. These two guys, Watson and Crick, they had been studying the structure of DNA for a long time, and they were trying to figure out what it looked like, but they couldn't figure it out. But as soon as they saw Rosalind Franklin's picture, they instantly knew they'd been working with it long enough that that picture meant something to them. And they were able to go, I know what it looks like, and they were able to create a model. And they knew that it was the double-stranded helix. Okay? So remember Watson and Crick because they are credited with discovering the shape of DNA. Okay? We know that DNA is a double helix, double because it has two strands, and helix because it is twisted. Okay, helix is a twisted shape, like a ladder that twists around, one of those staircases. Okay, so it's connected like a ladder, and then it's like you twist the ladder. And we'll learn more about this as we keep going, what all these little pieces are. So let's study some of our DNA vocabulary that we need to know. First, we need to know gene. Gene is a segment of DNA that codes for a specific protein. DNA has long stretches known as introns and exons. Introns are stretches of DNA that do not code for genes. They're sort of extra DNA. We're not really sure why we have it. Some people believe that it's evolutionary leftovers, that they used to be genes for our ancestors, but that they were no longer beneficial, so we stopped coding for them. Some think that it's sort of a fail-safe, so that in the future, if there's something in there that could be helpful to our evolution in the future, that it could be activated and become genes. But nobody really knows 100% for sure why we have introns. The other stretches of DNA are exons. These are the stretches of DNA that do code for genes. So the long stretches of exons actually have genes in them, codes for how to make specific proteins. Now these words you got to be careful with because they all sound really familiar, but they all are different. They're different portions of the DNA or DNA in different stages, okay? So make sure you take these down. These are all word wall words, and make sure that you know the difference between them. So the first one, chromatin, this is when the DNA is all loose. This is most of the time in your cell. It's loose so that it can be read easily and used. It kind of looks like a bowl of spaghetti. Then you have a chromosome. When your cell gets ready to divide to make more cells, we got to make sure the DNA gets divided correctly. So to do that, we got to make it less messy. So it tightens up and it tangles all up and wraps all up on itself so that it's as small as it can be. And when it does, it takes on the shape of an X. And this is called a chromosome. Okay? A chromatid is 1/2 of the chromosome. So it's 1/2 of the X. Then you have telomeres. Okay, notice the pronunciation of that one, telomere. These are the ends of the chromatids or the ends of the chromosomes. They're the tips of the X. And these are actually a redundant set of bases, and they act as protectors because every time you copy your DNA, sometimes you stop copying a little early and you cut off the tips of the chromosomes. So these are big, long chains that don't actually code for anything. So if you lose them over time, it's okay. And some scientists say you can even determine the age of a person by the length of their telomeres. So that's pretty cool. And finally, you have the centromere. This is the center of the chromosome. So this is where the X comes together. And this is important because during cell division, which is our next unit, this is where the centrioles are going to attach to the chromosome. Remember, those centrioles are organelles that are only found in animal cells and are used in cell division to rip chromosomes apart. Here's a picture representation for you. So this whole thing is a chromosome because it's X-shaped. 1/2 of the chromosome is a chromatid. So for every one chromosome, you have 2 chromatids. The very tips are the telomeres. So for every one chromosome, you have 4 telomeres. And then for every chromosome, you have 1 centromere, which is the center. And the last thing we need to talk about today are plasmids. Plasmids are special circular rings of DNA that bacteria can use. Okay, it's a circular strand of DNA. It only contains 1 or 2 genes in it, and bacteria can trade and share them. Bacteria can also pick them up out of the environment. So if you kill a bunch of bacteria on the counter, but another bacteria comes along, it can pick up plasmids that were leftovers from the bacteria you killed. Now we can convince bacteria to help us out. So the really big important one is insulin. We can make bacteria make human insulin because we take the human gene for insulin and we put it in a plasmid, a circular ring of DNA, and then we give that plasmid to a bacteria, and the bacteria will read the DNA plasmid and it will make human insulin. This is so important because people who have diabetes, they can't make insulin to control their blood sugar, and so they have to inject it. Used to, we would take pig and cow insulin and people would inject that into themselves, but that had a lot of risks because a lot of people were allergic to it because it's a little bit different than human insulin. But now we can make bacteria make human insulin for us, which is really safe and really cheap. So that's really cool. The next activity that we're going to do is going to involve learning more about how this is done.